Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Have you ever wondered how scientists figured out that invisible microbes, not bad air, were causing deadly diseases? Today, we are diving into the story of Louis Pasteur, the brilliant mind who defeated the bad air theory and changed medicine forever. From developing vaccines for deadly diseases like rabies and anthrax to inventing pasteurization to make our food safe, Pasteur's discoveries revolutionized the world of microbiology. But who was this man? And how did his experiments uncover the secrets of germs? In this video, we will explore his life, his groundbreaking experiments, and the discoveries that earned him the title the father of microbiology. So get ready because the story of Louis Pasteur is as fascinating as it is life saving. So, coming to the introduction, he was a French chemist, and of course, he is known as father of microbiology. And his research established the role of microorganisms in fermentation, spoilage, and infectious diseases, thereby laying the foundation of medical microbiology. Then explain his major contributions to microbiology. So first thing, study on fermentation. Before Pasteur, fermentation was believed to be a purely chemical process. Then Pasteur observed few things. He demonstrated that fermentation is caused by living microorganisms. It is not a chemical process. He showed that, I mean, of course, it is chemical process, but not purely chemical process. It is caused by living microorganisms. So he showed that yeast causes alcoholic fermentation and bacteria causes lactic and acidic fermentation. He proved that specific microbes produce specific types of fermentation. Then what is the significance of this? This basically established the concept of microbial specificity. It formed the basis of food microbiology, dairy industry, and brewing and wine industries. The second contribution is disproof of spontaneous generation. The theory of spontaneous generation stated that life arises from non-living matter. It's now it finds like it's baseless, but at that time everyone believed that. So in order to disprove it, he made the Swannick flask experiment. Now see the diagram there. So first we apply the heat. There is broth was boiled in flask with long curd next there. So heat was applied and it was let flask set, but there was no bacteria present after it. But when we remove the neck and let it set, the bacteria was present. So basically, this long curved neck, there air could enter, but dust and microbes were trapped. So broth remained sterile indefinitely, and when the neck was broken, microbial growth appeared. And coming to the conclusion, so microorganisms originate from pre-existing microorganisms. Life does not arise spontaneously. If life was arising spontaneously, then even when the neck was not removed and it was let to sit, the flask was let to sit, the bacteria should have been originated, because if it was originating from non-living, there could also be bacteria, but it didn't happen. So that basically disproved the spontaneous generation theory. Next, coming to the germ theory of diseases. In the title we said the man who defeated the bad air. So this is the basic theory, that is germ theory, which basically defeated or which basically disproved the miasma theory. The miasma theory basically said that diseases are caused by bad air or vapors from decaying matters, from foul smelling, from sewage, all those things. The air which was present, that was causing the diseases. Even if you observe the name malaria, it basically says mal area, mal, bad area, air, bad air. But it's not the case. So Pasteur postulated that, see, microorganisms are the cause of infectious disease, and it is the diseases are not caused by bad air or vapors. So that he proved with the scientific basis, and he gave the scientific basis for aseptic techniques, antisepsis, sterilization, and infection control. So germ theory is the cornerstone of medical microbiology. Okay. And next, coming to the development of sterilization techniques, he introduced the principle that heat destroys the microorganisms. So he gave contributions like controlled heating to eliminate microbes, and he laid the foundation for the development of stem sterilizer, hot air oven, autoclave. Now all these things, which are the sterilization techniques, we are going to discuss it in another video. So stay tuned for that video also. And coming to the importance in microbiology, it basically what is the importance of sterilization techniques? It helps prevention of contamination and the maintenance of pure cultures and safe laboratory practices. And then pasteurization of milk. So this is, if he didn't give the pasteurization of milk, I don't know, like probably someone else could have invented after a few time, but now it has become so important process in our daily life. So heating milk at specific temperatures for a fixed time and followed by the rapid cooling. What are the benefits of this? See, it destroys the pathogenic organisms and it prevents milk-borne diseases such as tuberculosis and brucellosis. Now many of you may ask, wait, is tuberculosis is a milk-borne disease? See, the spread of tuberculosis happens in a variety of way, and here in milk-borne there is something called as Mycobacterium bovis, which belongs to the Mycobacterium family itself, and it is present in the milk of the. It basically present in the infected cattle, so it may present in the milk, and if you don't do the pasteurization and if you drink, you can get it. And brucellosis, there are some microorganisms called as melillensis or Brucella abortus. So this Brucella melillensis, it is present in the goat and sheep, and if you drink the milk of goat and sheep without pasteurization, you can get brucellosis. And Brucella abortus, it is present in the cattle, so there also you can get it. So pasteurization prevents all the chances of getting diseases through the milk. Okay, and it does not alter the nutritional value significantly, and pasteurization is a major public health measure. Then contributions of immunology and vaccine development. So Pasteur introduced the concept of attenuation of microorganisms. What is attenuation? You basically weaken the bacteria and give that bacteria, like for example, if not only bacteria, it can include a variety like viruses also, all the, that's why we mentioned as attenuation of microorganisms. So consider rabies virus. Now you basically weaken that virus and you again give it, because now that you remove all the virulent factors, so it cannot cause any pathogenicity, but because of that our bodies will develop, our body becomes prepared for that virus if it comes in future. So that's how vaccines work, and we will study about this in more in immunology. But yeah, he introduced the concept of attenuation of microorganisms. So he developed vaccines for fowl cholera, anthrax, and rabies. You can easily remember this by using a mnemonic CAR. So C stands for cholera. Of course, you have to remember it's fowl cholera. A for anthrax and R for rabies. Okay. Then coming to the importance, established principle of vaccination demonstrated that weakened microbes can induce immunity, and he laid foundation for immunology as a science. And then liquid culture media concept, Pasteur used nutrient broth to grow microorganisms, and he introduced the concept of liquid culture media. We're going to study variety of culture media in upcoming videos, but the basis was laid by Pasteur. So significance allowed cultivation and study of bacteria, and it was essential for fermentation studies, vaccine development, and microbial growth analysis. And the last but not the least, it is institutional contribution. He founded the Pasteur Institute of Paris, which became a global center for microbiology research, vaccine production, and training of microbiologists. So that's all about the Louis Pasteur, the father of microbiology. I hoped you enjoyed studying about him. And if you found this video useful, please like and comment and show your support. And yeah, for exam, they have asked Louis Pasteur questions. So you basically have to write his contributions to the microbiology, because not more about his personal life. Just give an introduction and write all these contributions and explain and draw that diagram. You are good.